Last week, I had you guys look at mutations, and I saw a lot of people really struggled with it. So we're going to take some time today to go over mutations and wrap up. So the big ideas, you should know what the two main categories of mutations are, what kind of things cause mutations, and then what can be the result of a mutation. We tend to think of mutations in terms of sci-fi, so we definitely want to talk about the real science of it instead of the weird stuff that you guys are probably imagining in your heads. So if you're not sick of this yet, let's take another look. Central dogma: DNA to RNA to proteins, right? Okay, so DNA acts as our instructions for making these various proteins. And last week we looked at a DNA sequence right here that then can be transcribed into RNA and translated into a protein. And the protein sequence that we ended up with from this DNA sequence here was miss ya, stay safe. Okay. So when we are looking at mutations, we're looking at any kind of change to the DNA or the RNA. If we change the DNA or the RNA, then that potentially means that we could affect the protein that results at the end. Now, when we look at mutations, we're going to be looking at two main categories, and it's important to understand all of this in context. Remember that your DNA sequence is your sequence of these DNA base pairs right here, right? So let's say one strand is TAC, TAGT, etc., etc., etc. Eventually, that entire sequence that makes one single protein makes up what we're going to call that gene. And then the DNA strand, which is super duper long and has tons of genes on it, is then going to coil really, really tight to form individual molecules of DNA along with all kinds of proteins that are associated with it to help it coil. And remember that these are called chromosomes. So in this example here, this little band right here might represent this huge DNA sequence. So kind of keep in mind the scale that you're looking at. This would just be one individual chunk of the DNA that codes for one protein, so this would be just a single gene, whereas chromosomes have potentially thousands of different genes on them. And in humans, we have 23 different chromosomes, all with a different assortment of genes on them. So we'll start out by looking at one category of mutations called chromosomal mutations. And because there's so many genes on a chromosome, if you have a chromosomal mutation, it will affect a lot of genes. Bigger mutations lead to more genes being affected. And so here is an example of just a chromosome. This is what we expect it to look like for this particular example. And there's a whole lot of different ways that that one chromosome can be mutated. So let's go through and look at all of them. We can have a deletion. A deletion is where part of the chromosome is deleted. It's missing. So we've lost the genetic information that is contained in whatever section is lost. A lot of times deletions will be pretty substantial, but yeah, we won't go too much into those. In addition, you can have, just like a deletion can happen, we can have a duplication. So notice that we've got now, whatever these genes would be in this area are duplicated. We've got two of them, and maybe that doesn't matter, and maybe that has really huge consequences. It depends on what the genes are. We can have an inversion where part of the chromosome just flips around, and it doesn't seem like this would have a very big impact because you're still left with the exact same genes. But there might be something up here, for example, maybe in this yellow portion that controls. Here, let's look at this one over here. Maybe something in this yellow portion controls a gene right here and says, ramp it up, make a ton of that protein product. Well, if you flip around parts of it, now that yellow part is still saying, make a ton of the protein, but you're instructing a different part of the chromosome to ramp up production. So, you know, inversions can definitely have a pretty big impact. This right here doesn't even belong on this chromosome. So when part of one chromosome is mixed up completely with a different chromosome and you get two parts that kind of combine together, we call that a translocation. And then finally, you can end up with an entire chromosome that during the process of cell division, when the chromosomes are supposed to split apart, they just don't. And these are going to be called non-disjunction. Some examples of non-disjunction are things like Down syndrome, which is going to lead to extra copies of entire chromosomes and all of the genes that are on them. Okay. In addition, right, so if chromosomal mutations involve a whole bunch of genes, we can have much, much, much smaller mutations that really just affect part of a gene. And those are going to be our point or our gene mutations where just one or a couple of nucleotides will be affected. Sorry, I gotta block through these guys. There we go. So when we're looking at point mutations or gene mutations, we're just looking at, like I said, one or a couple of nucleotides that get changed. So maybe an A becomes a T or something like that. And so we can have a substitution, such as what I just mentioned. We can have an insertion where an entire new nucleotide pair is added just somewhere within the DNA strand, or conversely, deletion, where we now get rid of a nucleotide. And the results of a substitution can be really varied. With substitutions, we can have those silent, those missense, or the nonsense mutations. Insertions and deletions both tend to have the same consequence, which we're going to call frame shifts. So let's take a look at these, because this is really where a lot of people struggled. So let's say that this sentence represents our, our DNA sequence, right? And each set of 3 nucleotides, obviously A's, T's, C's, and G's don't make big sentences, so I'm taking some creative liberties here, but each set of 3 codes for a particular word. So our protein that we're producing is the cat and the dog ate the rat. Okay, that just makes it a lot visually easier to see what's going on. If I have a substitution mutation, that means that I've taken one letter and changed it out for another. In terms of DNA, that means that whatever my DNA code was, I have a new DNA sequence, but it's still coding for the same amino acid. So for that reason, it is silent. Here's your example here. Changing one letter doesn't change in the meaning at all. Everything's exactly the same. The cat and the dog ate the rat. A missense mutation is going to happen when one amino acid gets substituted for another because of a change in the DNA. So for example, here, instead of dog, we've got the cat and the dot ate the rat. A little bit of changing of the meaning, but not necessarily huge. And the results of this, the consequences, can really vary. And these are by far the most common types of mutations that you'll see within single genes. You can also have a nonsense mutation. So instead of coding for a different amino acid, it codes for a stop codon. Remember, that's kind of like your period. That's going to end the amino acid sequence. The cat end. So instead of an A, it got substituted with an E, which caused the rest of the sequence to stop. Okay. Here's the one that a lot of people got confused on, so I want to clarify. Insertions and deletions do just mean that. You're adding or taking away one letter, one nucleotide. But the protein that results is going to be hugely affected. If I were to take away just one of these letters, don't forget that the reading frame, the way that I read, or the way that our bodies read DNA and RNA, is by reading it 3 letters at a time. So if you get rid of one or add one in, the entire thing shifts down. For example, if I got rid of A, I no longer have the cat and the dog ate the rat. Now, all of the letters shift down one so that I'm still reading in groups of 3. The kute, nt, head, ota, tet, her, at. my meaning has changed pretty substantially. And if these frame shifts happen later, they don't tend to have as much of an effect. But if they happen really early in the gene, near the beginning, then you can imagine that those are pretty darn detrimental. So here are the examples that I gave you. Make sure that you're really understanding now, looking through and realizing why each of these is what it is. And we can see here that the deletion with what started as the polypeptide being miss ya, stay safe, all of a sudden I have huh? Anyway, okay. So enough talk about mutation. What does it matter, right? Like that's really the question that you want to know. Is this mutation going to lead to like a cool new organism with like extra arms and weird toes? Well, it depends. So a lot of mutations are out there. In fact, every difference between you and me is because of a mutation somewhere along the line. So we can look at mutations, and a lot of them are associated with diseases. So for example, it just takes one amino acid change in the gene for hemoglobin to go from having a normal blood cell, a red blood cell that looks like nice and round, to what is very sickled, kind of like the big chopping thing that farmers use to cut down wheat is a sickle. So a sickled cell is one that kind of curves over like this. And as you can imagine, this sickled blood cells, they don't really travel through the blood very well, through the blood vessels. So sickle cell disease is a prime example of a mutation that probably isn't so great to have. But as we'll see later, we'll see how far we get. Sickle cell can actually be a really good thing depending on the environment you're in. As it turns out, people with sickle cell cannot get malaria, which is a bloodborne disease that is carried by mosquitoes. So if you live in an area where malaria is really common, then having sickle cell disease sucks, but at least you can't get malaria. So there's kind of a cost and benefit. A lot of mutations have to do with controlling when the cell divides. And a cell that divides too fast is cancer. Spoiler alert. So a lot of cancers are now being identified as, like, scientists are finding the mutations that lead to those particular cancer. So for example, you've probably heard of the BRCA1 gene or the BRCA2 gene. Those are the ones associated with breast cancers. We can also have, so here's a diagram of the eyeball, and in the back of the eye we have these different rods and cones, and they pick up light waves. If you have a mutation in the cells that pick up colored light, it leads to color blindness. So that's just a mutation. But not all mutations lead to something that's harmful in some way. Our ancestors all had brown eyes, and so a mutation that in the gene for pigment, for the brown pigment, leads to a loss of brown pigmentation in the eye. This isn't necessarily harmful. It's not beneficial. It's just different. So if you are blue-eyed, know that you are a mutant, just like the rest of us. We all have something mutated, but that's an easy one to visualize. Things like how we can taste are also examples of mutations. One of my favorite examples is the reason I hate cilantro so much, but other people really like it. I have taste receptors, so I have a gene for a taste receptor that picks up a chemical in the cilantro that makes it taste like soap. So if you eat cilantro and it doesn't taste like soap, the reason is because that taste receptor for you, somewhere in your genetic lineage, was mutated so that now you can't pick up that soap flavor and all you get is something, I've been told, that's really good. And in fact, our metabolism is also controlled by our genes, and you can have different mutations that lead to changes in how we metabolize food. Sometimes for the worse, but sometimes for the better. For example, our ancestors couldn't drink milk into adulthood. The reason is because there really was no reason to. And as Europeans relied more and more on cows for their milk and for their nutrition, People that had weird mutations that would allow them to continue breaking down lactose into adulthood, they had a benefit. And so that mutation continued on, and that is what led to people who could tolerate lactose. If you don't have that mutation, then you are lactose intolerant, and that means you can't really digest milk comfortably. So that is a good example of a time when a mutation was really beneficial. And as we move into the next unit, this is all going to tie into variation of humans and of other organisms, because all variation within a population, within a group of organisms of one species, all variation is due to some kind of mutation that has happened. And we're going to refer to different versions of a gene, which is a result of mutation, as different alleles. So your assignment is to now take the quiz that is up on Schoology. You have unlimited attempts. You will not get to see what you got on the various attempts. So if you don't get 100, you can go back in, retry it. It will be based on your highest score, so you can take it 1,000 times if you want. If it takes you that many, oof. But yeah, anyway. So if you've got questions, let me know, but hopefully I've clarified a little bit. I miss you. Be good and have a good start to your next week.